The global trade in plants has had the unintended consequence of spreading pests and disease around the world, damaging ecosystems. Against the backdrop of a warming climate and the need to ensure our crops are resilient to climate change, we must also increase the function of our forest ecosystems to effectively sequester carbon dioxide and to enhance global biodiversity. It's clear we need new tools to detect and protect and mitigate against both invasive pests and diseases and those that are of importance to both food production and the wider environment.
Through the Bacterial Plant Diseases Programme, supported by a number of funding organisations, Niab and the Birmingham Institute of Forest Research obtained funding to support research to address bacterial threats.
Pseudomonas is a remarkable plant pathogen. It can cause disease on many different wild plants, as well as food crops such as kiwi, beans, tomatoes and cherries. We've been studying cherry pathogens for about 8 years now and have discovered that different Pseudomonas strains have independently figured out how to cause disease on cherry, each time using a different set of proteins and toxins.
What we don't understand is how specialisation onto a new host evolves in the natural environment, how frequently it occurs, and how populations of Pseudomonas vary over space and time.
Working with cherry growers and landowners across the UK, we're sampling naturally occurring Pseudomonas bacteria from the orchards and natural woodlands to understand how much diversity there is on cherry and related species, and whether any of the strains that we find have the potential to cause disease.
Our previous work has shown that gene exchange, the transfer of important gene related to disease-causing ability from one strain to another, may be mediated by mobile genetic elements. Work in this project is testing whether this is the case and where in the environment this genetic transfer can occur.
We have a wide array of tools that we can use to study which genes are important in the bacteria for host specialization. We can train machine learning models to associate the complement of genes a pathogen has with its propensity to cause disease in a particular host. With the many thousands of genomes that we're sequencing in this project, we will have a great data set to address this challenging question.
Combining molecular biology and machine learning has the promise of developing new tools that scientists and policymakers can use to enhance the speed and the level of detail that they can apply to decisions about what effective disease prevention and mitigation strategies might be. Longer term, this information will be invaluable to plant breeders who can make more informed decisions about disease resistance breeding.
And for growers, we hope this work will also underpin advances in targeted biocontrol approaches, with disease control tailored to the local bacterial community and pathogen, providing solutions that both boost productivity and do not negatively affect the environment.